In the previous video, we focused on how DNA is encoded into messenger RNA during transcription, where that takes place in the nucleus, and one of the strands of DNA is read, transcribed into messenger RNA, which we can see right here, and that mRNA leaves the nucleus and then is going to be used during translation to focus on, to build a protein. So this video is focusing on this second step, okay, translation, how we go from messenger RNA to a protein. It's very important at this time to remember that the building blocks of proteins are amino acids. So remember when I said back in September that you can't forget this stuff, it's going to come back to haunt you later on? Here it is again. So we need to make a very large complex structure, protein, out of these individual amino acids. Now there are 20 different amino acids, and what makes each amino acid different, if you recall, is this variable group. So we have, over here we have the amine group right here, then this is the carboxyl or acid group, a single hydrogen, okay, bonded to a central carbon, and then the variable group. So we can see the variable groups in here. These are all of the parts of the 20 different amino acids that actually make them different, okay? And so on and so on. You can see some of them are much more complex than others. So the basic part of the amino acid is the same, but these variable groups change the structure of the molecule and change the shape that it takes on in the end. So when we're looking at protein synthesis and translation, what we're concerned with is how does the messenger RNA that was made during transcription from the DNA determine what the order is of all of these amino acids. So we can see these different amino acids here, all in a different order, and that sequence determines the protein and its function. So let's look at how that happens. As you hopefully recall, there are 3 major types of RNA that we are concerned with, and all of these are involved in translation. So we have the M, messenger RNA, T, transfer RNA, and then we also have R, ribosomal RNA. The ribosomal RNA is an important part of the structure of the ribosome. Remember, ribosomes are the location or site of protein synthesis, those tiny little bodies that are found in all cells that are responsible for translation. They're responsible for reading the messenger RNA and assembling the amino acid or polypeptide chain. And let's focus on what mRNA and tRNA do in this process. So here's our mRNA that was made during transcription. And you'll notice that the mRNA, we've divided up the nitrogen bases into groups of 3. So we can see right here. These are called codons. The 1, 2, 3, 4, 5, 6, 7. That's just showing you that in this sequence of RNA right here, or mRNA, we have 7 different codons. So there are 21 nucleotides, ribonucleotides, or 21 nitrogen bases, but because we're dealing with them in groups of 3, we have 7 different codons. Each of these codons, okay, so here if we look at another mRNA sequence, right, each of these codons corresponds to 1 amino acid. So we can say 3 nitrogen bases equals 1 codon, which equals 1 amino acid. What we need to focus on now is how these codons, these groups of 3 nitrogen bases, correspond to an amino acid. And that's where the tRNA comes in. Here's our tRNA molecule. Remember, it's kind of a little clover-shaped RNA, unlike the single-stranded long mRNA chain. There are 2 important parts of a tRNA, a transfer RNA. The first is down here. Notice this is called the anticodon. The anticodon is the opposite or complement to the codon. So it's complementary to a codon, and it's going to follow the same base pairing rules that we looked at before. So if we have a G on the anticodon, that's complementary to a C on the codon on mRNA. U would go with A, C with G. If there's an A, that would go with U. Because remember, there is no T, no thymine in RNA. On the other end of the tRNA, we have an amino acid. So we have the building blocks of the protein that we're going to be making. So the anticodon on one end of the tRNA corresponds or matches up with the codon on the mRNA, and with that comes an amino acid. So each tRNA carries one amino acid. So one tRNA, one amino acid. And what you can see happening over here is, so this right here, this little structure, brown in this picture, is a ribosome. So if you imagine step one, right, we have a tRNA coming in, and so here's the anticodon made up of UAC, match up with AUG. AUG, you'll see this a lot because that's known as the start codon. We'll come back to that in a minute. And it has an amino acid, methionine in this case, on the other end. You can see our next mRNA codon, CCG. And here's the tRNA, GGC, with the complementary anticodon. So that's going to come into this spot. And then what happens is we get the two amino acids joining together. So we start to see that chain. And then the ribosome shifts this way, so it would go to our next codon, UAU, and a third amino acid would come in. Here we have another view of the same thing happening. So we have our ribosome, we have our mRNA, and the codons. Every 3 bases on the mRNA is a codon, right? So we hear codon, codon, etc., and that would continue going. And then we have the tRNAs coming in, each with their own amino acid at the end, and we can see this growing protein. Now, a couple things I want to add on. It says a chemical bond forms. This specific bond is known as a peptide bond. And you might recall that from back in September. So the bond between 2 amino acids is a peptide bond. So this growing protein is also sometimes called a polypeptide, right? Many peptide bonds. So if you ever see something referring to a polypeptide, it's a fancy way of saying a protein or a chain of amino acids. So this continues going and going and going until eventually there's a signal that says stop. That's the end of this gene. That's the end of the protein that we're making, all set with translation. Now this funky looking chart right here is known as the Universal Genetic Code. And it's universal because it applies to all organisms, humans, bacteria, fungi, your dog at home, whatever it is, that tree in your backyard. Every organism uses the same universal genetic code, which basically says, how does the mRNA, the code on the mRNA, correspond to an amino acid? So let's look at a few examples. I mentioned AUG before. So if we have the code on AUG, what we're going to do to find out what amino acid that corresponds to is use this. I like this chart because it's a wheel and it kind of works its way in to out. But sometimes you see other charts that are more like tables. We'll say first letter, second letter, third letter. So we always start on the inside. So our first letter is A, second U, third G. Okay? So AUG, right? AUG corresponds to the amino acid methionine. This is also, AUG is known as the start codon. And it's the start codon because it signals where translation is to start for each amino acid chain. So every protein, the first amino acid in that protein is going to be methionine. If we look at a second one, I'm just going to pick CCA, okay, randomly. So if we look CCA, you'll notice that anything that starts the first 2 letters, CCU, CCC, CCA, CCG, all correspond to the amino acid proline. Now, why don't you try to do GAC? Do that one on your own. Take a moment. And hopefully you have found out that GAC corresponds to the amino acid aspartic acid. So you can see this is how you can use this universal genetic code. Now, something else that you may have noticed on this universal genetic code is that there's one word that appears in here that doesn't look like an amino acid because it looks way too familiar, and that's stop. So we do have 3 different stop codons, just like we have AUG as the start codon, saying, hey, this is where it starts. The 3 stop codons, which you may have figured out, are UAA, UAG, and UGA. You'll see all 3 of those correspond to stop. That's not an amino acid. That just says translation is all done, stop, and what happens is the ribosome basically lets go of the mRNA, and then that long chain of amino acids, that polypeptide chain that has just been made, is going to do one more thing before it becomes a functioning protein. But it has all of its pieces then. So that amino acid chain made up of all of those different amino acids, a long string of them as directed by the mRNA, okay, which initially was directed by the DNA, right? What happens is because all of those amino acids have different chemical properties, they interact with each other in different ways. Some are attracted to each other, some are repelled from one another, kind of just like magnets. So we get different bonding patterns that happen. So it's, imagine taking this long chain of magnets almost, and they kind of naturally will fold up. So we can see that long chain, all these little bumps represent amino acids, and they start to fold up until you get this different structure that's much more complex. If you think back to enzymes, which are proteins, right, so made in this way through transcription and translation, that protein has an active site where the substrate for the reaction is going to fit. So they're very, very large compared to most other things that we've been talking about so far. So we say, sometimes you might hear about the primary structure of proteins, which is the sequence of amino acids. Then how those initially fold up is known as a secondary structure. And then that secondary structure, all of those folds interact with each other to get a tertiary structure. And then many proteins are made up of multiple chains of amino acids that are all folded up together, and that makes, it's called the quaternary structure. So something like hemoglobin, as an example, is made up of 4 different polypeptide chains, or 4 chains of amino acids that are all folded up together and then interact to make the protein hemoglobin, which is responsible for transporting oxygen in your red blood cells. So in the end, remember, central dogma, DNA codes for mRNA, mRNA codes for proteins. So protein synthesis is involved with transcription and translation. DNA goes to the mRNA, transcription. The mRNA then goes to the ribosome, where the tRNAs are going to bring the correct amino acids and line them all up. And then that amino acid chain folds up to become a functional protein. So now we can start to think about how all of those changes that may occur in DNA may or may not change the amino acid sequence, and then that may change the protein and therefore change the function of that protein. And that's what we'll get into next.